Thanks for your kind introduction. I'm Ruyi Chen, a third-year PhD student at University of Queensland. And today I'm going to present my recent work about using the multimodality model to improve the zero-shot prediction of disease effect on proteins. So the first question is how to predict the mutation effect on proteins. And this is the answer. In the past several years, and different teams train different model based on different algorithms. This model collects the protein sequences or protein structures or some multi-sequence alignment. It contains some homology features, and they use the advanced algorithms and to train the model to predict the mutation effects. But our question is there any other information can be further used to improve the disease effect? So we got some ideas from the natural language processing. Just as the English and the German, it can convey the similar ideas. Some of you may understand the differences between the English and German, but I can't. I just use it as an example. So there would have some differences between the two languages. The genetic information also has this kind of translation from DNA, mRNA to proteins. So our hypothesis is if the languages model training at the equivalent region, that is coding regions, but expressed at the different languages, say DNA and proteins, it may also have these linguistic characteristics. If this is true, if we combine these two languages model together, can improve the disease effects predictions. So for our experiment design, we collect the single point mutation from the ClinVar database, and then this missense mutation can be further divided into likely pathogenic, likely benign, and we combine the likely pathogenic into pathogenic variant. So it's more like a classification task. And then we choose this high quality missense variant and with at least one review start. And then we also choose the two different models. They are all transformer-based training via the mask language modeling objective. The first one is a codon languages model called the COM. It took wild-type CDN sequences as input and tokenized them into 64 different tokens. And the second one is a protein languages model called ESM2, and it takes wild-type protein sequences as input and tokenize them into 20 different amino acids. We used the formula here to calculate the effect score or the log likelihood ratio generated by the two models. And then our hybrid model is built upon the combination of these two effect scores. We used the Bayesian optimization to choose the optimal weight assigned to each terms. And finally, we find if we assign the equal weight to both terms, it will generate the best result. And this is our result. We could find the COM model alone, that is codon languages alone, can distinguish this missense variant, but this performance is not better than the protein languages model. And more importantly, our strategy, our hybrid model achieved a 3% increase in ROC, AUC in classifying this disease effect, and it significantly outperformed the two single sequence-based models. And besides, it also performed well in distinguished pathogenic missense variants. So for the next step, we want to know why we combine a PLM with CLM can improve these disease effect predictions. So for the first step, we identify those significantly different missense variant and that falling at these rejection regions. And then according to their model's performance, we further categorize this missense mutation into codon languages model better and the protein languages model better. Codon languages model better means it will assign a more negative value to the pathogenic variant, and it will assign a higher value to the benign variant, so that the model can distinguish this variant efficiently. And then we define or we calculate this bias ratio divided by the subset variant pair frequency divided by the overall missense variant pair frequency. If this score more than 1 means this missense mutation is enriched. If this less than 1 means it's not reached. And finally, we use a chi-square test to assess the statistical significance of these observed enrichment or depletion patterns. And this is our findings. This is a heat map, and the x-axis here means the wild type amino acid, and the y-axis here means this mutated amino acid. The color here means if they are enriched or not, and the star here means if they are significant enriched. So we first find the differences between the CLM, PLM predictions lies whether this mutation changing from the amino acid has a high codons, and so it have a tendency that it likely to change from the high codon residues to the low codon residues. And for those mutations that do not substantially change the codon usage, the effect score assigned by CLM and PLM are generally similar. Besides, we also find that the PLM and CLM differ in prediction-specific mutation types. For example, you can say the CLM is better at prediction the missense mutation like from T to M, while the PLM is better at prediction from M to T. It means the PLM and the CLM are complementary. So we have some insight that maybe in the future we are going to train a multimodality model to further enhance the disease effect predictions. And finally, I want to thank my supervisor, Michael Bowden, and all the members in the Bowden lab at the University of Queensland. I'm happy to take any questions. Thank you.
Thank you very much. Question from the audience? Okay, one question I would like to ask because, okay, sorry.
Hi, that was interesting. I wonder if you found that were very specific cases where the codon model improved over the protein level model. I wonder if a representation where you just use those codons.
Yes, actually, if you scan, due to the time limit, I didn't show the performance of the codon model alone to distinguish the nonsense or the synonymous mutations. In fact, it will assign the negative value, the more negative value to the nonsense mutations, and it will assign the higher value to the synonymous mutations. So I think codon languages alone is very sensitive to those mutation types. Yeah. So, but if you are interested, you can read our preprint. Yeah. So it have more details in it.
Thank you.
Other questions?
Very interesting work. Do you consider the secondary structure changes in the RNA in these cases while modeling it?
Sorry?
Secondary structure changes because of mutations? Secondary structure of RNA?
Second structure pattern? I don't understand, sorry.
Secondary structures of RNA?
The structure of RNA. If there is any type of base pairing, but I don't know if they are considering actually the whole. Are you considering the whole RNA when you do predictions, or just the protein sequence?
So actually, we just because our ontology is a language, so we didn't use structure informations. But if we want to further advance missense mutation or disease effect prediction, we should contain different kind of resources, as I mentioned before, like the multi-sequence alignment structure sequences. Here we provide the 4 different another resources, that is CDN sequences. So future it would be a more advanced model that can be further used to classify this mutation effect. And we show the model they are complementary, so it can maybe improved.
Okay, thank you very much, Ruyi, again.